Welcome. My name is Gaspard de Tournemire. I'm a postgraduate student in ecology and I've been interning at the French Institut de l'Agriculture et de l'Alimentation Biologique, or ITAB, for 6 months. I am the author of this short training unit, which is the first in a whole life seeding training module that is focused on the microbial life associated with organic seed production. This first unit acts as an introduction to the topic of seed microbiota. So I will first define what are seed microbiota and how they are assembled, how they come to be. I will indicate in these assembly processes which may be affected by seed growing techniques and plant breeding generally. Then I will describe a technique that allows for the analysis of the patterns found in these microbiota, and I will report results of 2 studies on crop seeds as examples. They will give us an insight into what we currently know about the general structure of seed microbiota. At the end of this module, you should thus have gained a general oversight of seed microbiota, how they are studied, and the types of results that can be obtained from seed microbiota research. The first subject we'll address is a characterization of the microbial life that is associated with cultivated plants, and more specifically, their seeds. As an entry in this complex world of plant-microbe interactions, we'll take the common bean, so Phaseolus vulgaris, as a model plant. Here is an illustration of its life cycle, considering the plant as an organism made up of cells sharing a single genome and structured in well-defined plant tissues. But these plant cells are not alone, and each phase of this plant life cycle is associated with a diversity of microorganisms which inhabit the plant structures. A distinction can be made between microbes surrounding the plant organs, which are known as epiphytes, and those that manage to spend some time inside the plant tissues, known as endophytes. As shown here in the center, seeds carry their own epiphytic and endophytic communities, which are passed from parent plants to progeny through a process called vertical transmission. Among the many tiny forms of life that are hosted on seeds, we can find bacteria, filamentous fungi, but also yeasts and oomycetes, and a wide diversity of microorganisms in general. This wide diversity of microorganisms transmitted by seeds can act mutualistically. They can bring many benefits to plant functions. Examples of this include nitrogen-fixing bacteria, which can live near the root systems of plants and promote growth by complementing the plant nutrient uptake, or endophytic fungi that live in the vascular system and can secrete toxic compounds that protect leaves from herbivores. These close interactions between plants and the microbial communities they host are the grounds on which stands the concept of a plant holobiont. This word symbolizes the ecological unit made up of a plant and its microbiota. This concept of a plant holobiont can be very useful to gain a more complete understanding of the microbial processes that are associated with diseases. A disease-associated microbiota can be considered as brought out of balance by an environmental stress, resulting in disruptions of the microbial community assembly processes. Plant health management can gain precious insights from the study of the seed as a holobiont. So to briefly summarize this first overview of the concepts underlying the study of the seed microbial life, I'll simply stress that seeds are closely associated with diverse communities of microorganisms. The structure of these microbial communities, known as a microbiota, is an essential dimension of plant health. Okay, now we'll go a bit further in this introduction and we'll take interest in the processes underlying the seed microbiota assembly, meaning how and from which sources can seed acquire their microbiota. And as a backdrop to this further presentation, we'll be again using the common bean plant life cycle from seed to seed in its own particular environment. Here again, we try to illustrate the diversity of microorganisms that share this environment with the bean plant. The first elements we can focus on are the various sources of microbes that are present in the plant's environment. Generally speaking, the plant can acquire microbial communities transmitted by the soil, by contact with neighboring plants, by passing animals or pollen grains, which can be dispersed by the wind, and or from rainwater, for example. Then, the microorganisms that have been brought on and in the plants from these sources are subjected to a selection pressure by their new habitat's microenvironment and to a specific control by the host plant, through chemical substances secreted by the plant or by the responses of its immune system. The seed-carrying plant thus continuously interacts with its microbiota's composition, and organic plant breeding may benefit from the consideration of these functional traits of selection. And finally, as mentioned before, seeds carry microbial communities in part transmitted by their parent plant. These microorganisms, they benefit from a priority effect in the assembly of the seedling microbiota, and they have functional implications in seed processes such as germination. Taking into account the seed microbiota thus emphasizes the major role of the multiplication environment on seed quality. To quickly go over what these last points imply, I'd simply state that the plant-mediated assembly of diverse microbial communities from the environment has a large impact on seed quality. And as another reminder that seeds are the main drivers of what is called vertical microorganism transmission, which is the type of transmission that occurs within a plant line from generation to generation. Now that we have a better idea of the importance of microbial life in seed health and quality, I'll describe a widespread technique used for studying microbial community structures on seeds. This technique, known as metabarcoding, relies on DNA extraction and sequencing to identify the microorganisms present in and around a seed sample and evaluate their relative abundances. To explore the method's strengths and weaknesses, I'll quickly outline its general steps. So first, a seed sample is soaked in a neutral solution. This allows for the sampling of the microbes that are present in the seed sample. Then, the DNA present in the soaking solution is extracted. It's important to note that this DNA extraction process does not ensure our analysis focus on viable microorganisms, as conserved DNA from dead organisms may be extracted as well. In this extracted DNA, the segments corresponding to marker genes specific to microbial kingdoms, such as bacteria, fungi, etc., can be amplified by polymerase chain reaction, or PCR, to reach a high enough number of copies of these genes to allow for sequence analysis. This amplification yields a large library of the copies of the marker genes present in the sample's extracted DNA. The sequences of these segments can then be read at a high throughput by a machine called a sequencer. After using informatic tools to clean carefully the resulting read sequences, we are left with a set of sequences deemed uniquely different from each other. These unique sequences are labeled as amplicon sequence variants, or ASVs. Comparison with an appropriate database allows for the attribution of a specific taxon to each unique sequence. A taxon can be as precise as a specific strain of a bacterial or fungal species, but it is sometimes limited to a wider taxonomic level, such as the genus or family. We are thus left with what's known as a table of abundances. It evaluates the abundance of specific taxa in each analyzed sample. From this table of abundances and the assigned taxonomy, we can compare the structure of the microbial communities of multiple seed samples depending on whichever condition we deem interesting. Here we can see basic analysis of the metabarcoding data for bacterial communities of 5 subsamples of seeds from the same variety and same harvest. We notice a high variability of taxa richness and community composition, as is given from this community profile on the right. So now, to summarize what's just been told in plain terms, we can say that the DNA extracted from a seed soaking solution can be processed to give estimations of which microbes are present on and in the seeds and in what proportions. This can be used to compare microbial community structures between samples, but it is important to keep technical biases in mind when analyzing metabarcoding data, as results may be overinterpreted. As a final message for this presentation, I'll go over 2 examples of work done in the field of seed microbiota research to show applications of the outlined metabarcoding method for the analysis of the processes described in the introduction. The first study I'll present is focused on the microbiota of single bean seeds. In the spirit of the composition profile I showed you earlier, the study produced a bacterial community profile of 144 individual mature bean seeds. To keep it simple, in front of this intimidating wall of color, we can first simply note that bacterial communities analyzed vary very greatly between seeds. The second interesting observation we can make is that the vast majority of bean seeds hosts one principal bacterial taxon, which makes up most of the identified bacterial reads. An interesting point to keep in mind from this study is that the distribution of the different microbial taxa harvested from one field, or even from a single plant, isn't homogeneous. The microbiota of seeds from the same seed batch are different from each other. There is a high variability of microbiota structure between seeds. In addition, we note that most seeds host a low number of taxa, with one principal bacterial taxon per seed, at least in common bean. The second study I will present now concerns a wider range of host plant species and larger sample sizes. Indeed, in 2021, a large-scale meta-analysis of seed microbiota data was published, and it can give us insights on the general patterns of seed microbiota structure across host species. Here are two other scary wall of colors from this study, but a similar message from previously. There is a very high variability in the structure of seed microbiota, although a few core bacterial and fungal taxa can be identified in a great diversity of plant species. At this scale, we can also observe a strong influence of host plant species and botanical family on the structure of their seeds microbiota. In this data, for over 1,000 samples of seeds of more than 40 plant species, patterns similar to those identified in single bean seeds can be observed. The bacterial and fungal communities for most samples are extremely variable in structure and characterized by one main ASV, or taxon, further emphasizing the selective nature of the seed habitat. And with this last remark, the first live seeding training unit on seed microbiota is over. So what can be remembered from this presentation? Well, first, we've defined plants as holobionts, closely associated with the microorganisms that inhabit their tissues. Seeds were identified as critical steps in the life cycle of plant holobionts. To gather information about the specificities of these seed microbial communities, metabarcoding techniques are presented, with emphasis on their limits to better understand the resulting data. With this in mind, we've observed from metabarcoding results that microorganisms hosted by seeds are very specific, with seeds offering a highly selective habitat. More generally, the complex patterns uncovered by the studies presented here have major implications on organic seed production. Considering seeds as holobionts themselves may be essential for truly robust plant breeding and seed multiplication practices. Thank you very much for your attention throughout this unit. I hope it may be of use to you, and I wish you a very good day.